All right, so see if you remember anything from last. Jeez, a long time ago, a week ago. What's different between first and second line of defense? All right, looks like you have some discussing to do. Look, we had at least one person vote for every answer. It's pretty good. That's ridiculous. All right, so maybe you need to have a little discussion there. All right, so what do y'all think? Everybody vote. Looks like we got 13, 37, 11. All right, so we had a big shift to F, which was B, C, and D. That's not right. The answer is actually B, which is what 30% of you put. So T cells, is that first, second, third line of defense? Which one is it? That's third line of defense, is T cells. What about B cells?
That's the first. No, really? I feel like that's the first. And then that's the second.
B cells are also third line of defense.
I give up.
I know, this part's hard. Specific, is that first, second, or third?
Which one?
Specific?
Third.
It is third.
It's on another.
It's a little mean because we didn't do third yet in detail.
It says right here, it's specific or cell or antibody based. Way in the beginning.
And then antibodies, first, second, or third? It is third. So whether or not immune cells are involved, it's the difference between first and second, which brings us to the Venn diagram that you're going to work on with your group there on the tables. Actually, not individual first, because we don't have time for that. So Venn diagram between first and second line of defense. You just got one thing. Which one has immune cells? Second, which one doesn't? So there's your first difference. All right, so maybe check in with your neighbors there, see if they've got something you don't. All right. So some similarities. What are some similarities? Non-specific. What else? They both protect you from infection. Sure. One thing that people always get it confused, locations. Where's the first line of defense? But like where in your body? Is it only outside? What about like your stomach acid? Or remember flow of urine was also?
So both would be inside, right?
Both would be inside, but only first is outside. So the first is inside and outside, the second is inside. So however you want to write that down. I think on tests, everyone always says the first line of defense is only outside of your body, but it's not. There's lots of places inside too. And then in terms of differences, basically, second line of defense is immune cells, first isn't. And then all the examples. So first line of defense, physical barriers like your skin or mucous membranes, chemicals like lysozyme, stomach acid, sweat, things like that. And then second, cytokines, inflammation, fever, interferons. Cytokines. Did I say cytokines already? I think I did. All right, so let's try this on your own first. It's a bunch of true-false questions. See what you remember. So as you're fixing those, go ahead and check in with your group mates there. All right, so first one. It's false.
Should be non-specific.
Should be not specific. What about B? Speeds up the response. C. That one's false.
In response to.
So you could say in response to viral infection or by virally infected cells. Either way would be right. What about D? Did I make all these false?
No.
Oh, good. E?
False.
That one's, yeah, it's false, not specific.
I changed it and I put the third line.
Or you could do that. Just have to be different.
I put the,
Very true.
That's totally fine. And what about F? True. And the last one? Very true. Extra true. Hopefully not too many of you put false for the last one. Be concerned about you. All right, so questions on the study guide.
a lot.
So many. I don't have one, so you have to tell me what it is.
I just have a problem with like when you say the B cells, there's different types of B cells. So like,
Like plasma and memory? We're talking about T cells?
no, I'm talking the B cells. There's different.
It says identify the function of the following blood cells.
Yeah.
And then it says
Oh.
B cells.
B cells make antibodies.
Okay, that's it. What about the T cells?
T cells?
Helper cells.
T helper cells kind of activate the immune response.
So they would activate B cells.
They do. So they're kind of, the T helper cells activate the immune response. Mostly like the specific, the third line of defense.
What's unique about each B cell and T cell?
What is unique about each B cell and T cell is their antigen binding receptor
Is it 'cause, like,
and their personalities.
Where they find like the B cells bind on the outside or whatever the T cells bind. Their binding receptor.
Just for E.
What's E?
Comes to the B cells and T cells.
What do you want for E exactly?
Basically, B cells make antibodies. T cells activate the immune response or directly kill infected cells.
Immune response.
It just says T cells in general, right, on there? Yeah. So,
Okay,
What did you say for the T cells?
so T cells activate the immune response or other T cells directly kill infected cells, like virally infected ones. That sounds like the same thing to me. So you said it on the outside of the cell and the surface?
the antigens right on the surface of the invading.
Oh, of the virus.
And then T cells can only connect to virus antigen on the outside.
Yeah, I would say technically that's the antibodies that the B cells make that do that.
Okay, so either
And he
way.
put antigen binding receptor.
That's all you need to know is the antigen binding receptor.
For what's unique about each one?
Yeah.
Or for B. No, they both.
No.
So what's unique about each of those cells is their antigen binding receptor, which you'll see kind of what that means later today.
So we don't really need to know what she just kind of said too? 'Cause
You will at some point.
that's not relevant.
Eventually need to know though, so like the B cells can go on the outside.
B cells can make antibodies that directly target the virus,
Darn B
And
cells.
T cells
and T cells can't. T cells deal with your own cells that are infected or broken.
Okay, that made a little more sense.
Yeah, that makes more sense.
B cells make antibodies and T cells do.
Yeah, they do everything else.
Yeah. T cells affect your cells, B cells affect the virus cells.
Oh my God.
What'd you say?
Cleared it all up.
T cells affect your cells.
Right.
And B cells affect the viruses.
There you go.
Which is helpful.
I put in that.
Yeah, me too. This whole chapter makes sense now.
No, it doesn't. Don't lie to us.
One little part of this makes
No,
sense now.
like when the tiny little part.
All right, so let's do part 2 quiz then. Nobody asked me about the first 2 questions.
I thought I knew it until you handed it.
Yep.
It's in my nose and I'm like, crap, what was that?
That happens to me on the test quiz too.
I think that's the first time. I think every other time I've done this, someone asked about those too. But anyways, antigen, type of cell, foreign molecule, molecule produced by B cells, a receptor on T helper cells. It is a foreign molecule. What is an antibody? We said it like 5 times. It's a molecule produced by B cells.
What letter was that?
That was C. Function of T cytotoxic. Produce antibodies, activate immune system, directly kill virally infected and cancer cells, or phagocytosis. It's the third one, directly kill the virally infected and cancer cells. What is unique about each B cell?
The binding receptor.
Antigen binding receptor. General function of cytokines. Bind to pathogens, activate immune system, kill pathogens. Activate the immune system. Right. First one was foreign molecule. All right, so let's see how far we can get on this one. This is not chapter 15. Don't worry. All right. So the third line of defense, it's adaptive and it's specific. Specific means it recognizes certain molecules and certain viruses or bacteria. Adaptive means you can get immune to certain bacteria. Like the next time you see it, your response is stronger. So you can like kind of adapt and not get sick from the same thing over and over again. And we'll see how that works. First up is our antigens. That's question one, some Play-Doh. Let's make this, see if we can make this snappy. So you're going to draw a big circle on your table and put 3 different shapes on the outside made out of Play-Doh. Right? And did it say it on there? So that circle you drew, that's like an invading virus or bacteria. So based on kind of what you did there, what do you think the answer is to those? All right. So it is E. It is all of this. So one of the important things I think that people always get confused on is your immune system isn't responding to like a virus or bacteria. It's responding to one of those shapes on the surface. So if they, you know, those like virus spreading into the world movies, they're always like, oh no, the virus is mutated, right? There's always that line in there somewhere. So that means that shape that they developed the vaccine or your body's fighting against changed shape. And now your body doesn't know how to fight it anymore. Does activate the immune system, and obviously, like you just showed me, there's multiple antigens on the surface, different shapes. All right, so next up, you're going to make some antigen binding receptors out of different colors of Play-Doh that bind to those antigens you made. All right, so everybody got those copied down? All right, so cool thing about the immune system, so you've got, how many different antigen shapes do you think there could be on viruses and bacteria? Not very many? In 3 dimensions? So it's probably like nearly infinite, because you've got, it's in 3 dimensions, first of all, and then size, and then, I mean, how many different 3D shapes can you make at a molecular level? Like almost infinite amount. So the challenge for your body is it's got to find a way to be able to recognize a nearly infinite number of shapes as foreign. So then what do? They make an infinite number of antigen binding receptor shapes. So that randomly, hopefully you have a shape that you made that fits one of those antigens. And you don't have to have one that fits every antigen, just, you know, at least one of the antigens that that virus or bacteria has, you recognize. And if it's a virus, it's got a decent amount. Bacteria have an enormous amount of antigens on their surface. So viruses are a little harder. Maybe they've only got 3, so hopefully you can recognize one of them, or else you're in trouble. So you'll see how that works. First little background, your B cells, They're made in the bone marrow. And then once they mature and they're ready to function, they go to your lymph nodes, which are all over the body basically, and then the spleen. Your T cells, pretty similar. They're made in the bone marrow, and they go to the thymus to develop. And then when they're finished developing, they go to the lymph nodes and the spleen. And that's mostly completed by the time you're like 15 or so. So you could take your thymus out now and nothing bad would happen to you because you're pretty much done making B cells. They're all in your lymph nodes and spleen already. All right, so when we say B cell development, one of the first things that has to happen is you make a whole bunch of B cells with different antigen binding receptors. So it's kind of a random process, the shapes they end up with. And you could see their little antigen binding receptors on the surface. And you could imagine, they could have all those shapes you made, those binding receptors. But you could imagine if you're making random shapes, do you think you could make a shape that would bind to your cells? If you're just making random shapes? You could, right? That would be bad. Now you've got autoimmunity, your body's killing its own cells. So before they're done developing, before you send them out into the body, to the lymph nodes, they're checked by special cells that kind of inspect these receptors, and they look for any that bind to your cells. So if they do bind to your cells, then they're told to kill themselves. So go into apoptosis, sorry, you developed not the right way, you don't get to live anymore. That's what happens. So they're eliminated. That's what these little X's are. Very important, because if you made B cells that attacked your own cells, you could die, basically. Or have your kidneys stop working, or who knows. So that's clonal deletion. After you do that, then those B cells go to the lymph nodes and wait for an infection to happen. All right, so which of these is true of the B cells that remain after that process? All right, so we're trying between C and E, and the answer is C. It's the only answer. And I put D on there because people know that B cells produce antibodies, but not right away. You'll see how that happens. It takes 5 steps to get them to making antibodies. And then the reason they don't make antibodies right away is that would be a real waste of resources. Because if you've got 100 million B cells all making antibodies, that's kind of pointless because most of them, those antibodies don't recognize anything. You only want to make antibodies against something that's trying to get you. Otherwise, you're just clogging up your blood with antibodies. All right. So what about the T cells? It's pretty similar. So the only difference is instead of developing in the bone marrow, they develop in the thymus. Does this slide look familiar? That's because it's the same process as the B cells went through. That's why we have the same picture. All right, so which of these is true of the T cells after clonal deletion? All right, so the correct answer is C again. And what did some other people put? E, so they don't recognize human cells at all. The ones that recognize human cells were killed in the deletion process. So only C there. All right, so in order to activate our B and T cells, we've got to get the antigen to the B and T cell. And the cell that does that is the antigen presenting cell. Most of the time, these are dendritic cells and macrophages. And if you notice, those are they in the first, second, or third line of defense? They're second. So you've got something infecting you. Those macrophages are trying to take care of it. They're eating those pathogens. Remember phagocytosis? But what if that's not good enough? What if you're not going to get rid of it just from macrophages? So they're able to activate the third line of defense. And the way they do it is they take antigens from that bacteria you drew and put them on their surface. So they can go show it to the B and T cells. Got a little movie of that, first 2 minutes.
After education within the thymus, T cells circulate throughout the lymphatic system and accumulate inside the lymph nodes, spleen, and other secondary lymphoid tissues. Other types of cells are also present, including B cells and macrophages, both of which can also act as antigen-presenting cells. Consider a macrophage. Should it encounter an antigen for which it has no specific receptors, it can still engulf it using, for example, its lectin-like receptors. This is termed nonspecific internalization. The pathogen bearing the antigen is contained within a phagolysosome in the macrophage.
So there's that lonely little virus that just got eaten.
Other vesicles fuse with the phagosome, bringing proteolytic enzymes
So this is
in.
like the steps of phagocytosis that you saw in part 1.
Elsewhere in the macrophage, major histocompatibility complex molecules are being synthesized at the endoplasmic reticulum. MHC class II molecules are transported by the vesicles, which intersect the pathways of vesicles released from the phagolysosome. Proteolytic fragments of the antigen bind to the MHC class II molecules in a special compartment, displacing the invariant chain of the class II molecule.
You don't need to know all these details, by the way.
Loading is facilitated by another MHC-encoded molecule called DM. The vesicle containing the MHC class II antigen fragment combination migrates to the surface of the macrophage, where the contents remain attached, presented to any Th1 cells that may be encountered.
So eat the virus, attach it to your little presenting receptor, and then put the antigen on top of it. And you can use that to go activate the specific immune system. All right, so how about with your group questions 3 and 4 on there? And remember, it's an APC, ate your bacteria that you made with the Play-Doh out there, and put one of the antigens on the surface. So we've got our macrophages, our APCs. And so next up is going to activate the specific immune system. So this is probably as far as we'll get with you. So that antigen presenting cell, it's going to go find a T helper cell that has that antigen binding receptor that you drew, and activate that T helper cell. So what does a activated T helper cell do? It divides and produces more T helper cells that has the same receptor. Simple enough. So you're going to draw that for me, which is number 5. So what we're trying to do is turn all those words into a picture. So read through that one, chat with your group, see if you can get it all figured out. All right, so everybody got those? And what those T helper cells do now is activate B cells that match the antigen, which we'll see in a little bit. They also release cytokines that turn on inflammation, and cytokines that activate macrophages, and things like that. And don't worry about the CD4 thing. That has to do with HIV. Because HIV targets those T helper cells because they have that receptor. All right, so we'll pick up with this tomorrow during lab time. Oh, and be a good child and put your Play-Doh away so it doesn't dry out.